First, that is the applications in agriculture. So in the three ways we can increase our agricultural productivity. First of all, that is agrochemical based agriculture. Then second is organic agriculture. Third is genetically engineered crop based agriculture. So in the biotechnological field, today's most useful technique, that is genetically modified organisms are useful in that we are modifying or altering the genes of the different types of microorganisms, right, like plants, animals, fungi, to get benefits or to increase the productivity and yield and the useful proteins present in that, right, to isolate it into the huge amount, right. First application in agriculture, that is advantages of genetic modification in plant. What are the different advantages, right? So first, it makes crops more tolerant to a biotic stress, salt and heat is the abiotic stresses to the plant, as the our daily environmental conditions are being changing. So due to that environmental conditions, our agricultural field is also get disturbed. So the temperature, and as we know, the rain is not happening proper time. So to overcome such problem, or to by this, the agricultural product is decreasing and the demand is continuously increasing. To fulfill that condition, we made the genetic modification in the plants. So what we have did, we have make the plant which are tolerant to abiotic stresses, right. Next is pest resistant crops. Why we are using that pest resistant crop? We are using this pest resistant crop to reduce the use of chemical pesticides, so the farmers can't get overwhelmed with the cost of the chemical pesticides, and it is also harmful for us to digest, and it is very dangerous for our health also. Third application is it helps to reduce post harvest losses. Next is it increases the efficiency of mineral usage by plant. It means it prevents early exhaustion of soil fertility. Then it enhances the nutrition value of food. Golden rice, it is the one of the example of nutritional value food, right. So here we are overcoming the deficiency of vitamin A in the developing country people by golden rice, which contains a huge amount of or large amount of vitamin A content, right. So the blindness disease is commonly happening in the developing countries. To overcome such problem, we are using the golden rice. Then GM, genetically modified crop, is used to create tailor-made plants to supply alternative resources such as high amount of starch, fuel, pharmaceutical to the industries, right. We will see to the next. Another application in the field of agriculture is the pest resistant plant. So as we know, the plant is get infected by the various insects and microbes, which is causing that plant diseased, and due to the diseased due to that disease, the our productivities get decreased. So to overcome such problem, but we are avoiding to spray the chemical pest resistance. So we are using the genetically modified plant. In that, we are using pest resistant plants which are act as a biopesticides, right. We are directly incorporating a pesticide, means resistant gene inside the plant's body. It also reduces the need for insecticides, right. We do not need to spray insecticides on this type of genetically modified plant. So next is the example. There are BT cotton, BT corn, BT rice, tomato, potato, soybean are the following example of genetically modified plant for the pest resistance. BT. Now, what we mean by the this BT? BT means it is one of the species Bacillus thuringiensis of bacteria, which is having a gene that is called as cry1 and cry2 gene, right? These genes produces a protein which is called as bt toxin, right? So this bt toxin proteins, when eaten or chewed by the various pests, so in the gut of that body, they will form a toxin and kill that insects, right? So what actually happening here is this bacillus thuringiensis proteins have the ability which is toxic to the insect. When insect eat the plant leaf or any part, so the Bacillus thuringiensis or Bt toxin is ingested by that particular insect. So in the gut, that is coliptrans, beetles, lepidotrans, tobacco, bedworm, armyworms, and dipterans, flies, and mosquitoes, it will get killed by the Bacillus thuringiensis or Bt toxin. So, in which form this Bacillus thuringiensis is present? So, Bacillus thuringiensis forms a toxic insecticidal protein that is called as Bt toxin in the form of crystals in a particular phase of their growth. But it does not kill the bacillus, means the toxin is produced inside the bacterial body, but it will not harmful to the particular bacteria, right? It is present in the inactive form, or we can call it as a protoxin. Yes. Next is this all process we have seen how this will happen, and this crystals is getting ingested by the particular insect, and it will go inside its midgut, and it will get it into the activated form, and it will kill or disrupt the midgut or intestinal part of that particular insect, and that will get killed by that bt toxin or that bacillus thuringiensis. Next, that is how this bt cotton is being incorporated into the plant body, right? Most bt toxins are insect group specific toxin, which is coded by a gene that is cry-I AC and named as cry gene. Each protein is encoded by the gene cry-I-AC and cry-2AB control the cotton bollworms that of cry-1AB controls the corn borer. So these are the genes or the crystals which is formed in the different parts of the plant, and which will infect the different insecticide pests, such as the cry-1AC gene, which will form the crystallized protein which is resistant to the cotton bollworm, right? When this protein, that is cry-1AC, is ingested by the cotton bollworm, that will get killed. Then second gene, that is cry-1AB, that controls the corn borer. Next is applications in agriculture. Another, that is nematode resistance in tobacco plant. So nematode that is named is Milidogene incognita infects the roots of tobacco plant, causing the reduction in the yield. So this is nematode which is decreasing the yield of the tobacco plant, right? Which is being prevented by the RNA interference strategy. What is this RNA? RNA is a method of cellular defense in all eukaryotes. It prevents translation of specific mRNA That is, it will silence the RNA translation due to a complementary double-stranded RNA molecule, right? So here is the stopping of translation process. So if there is no translation, there is no formation of a protein, right? So there is a in production of the tobacco plant. Next is pest resistant plant, and yes, another example of this is double stranded RNA, right? So which is it initiates RNAi and silence that specific mRNA of nematode, right? So this why we are using agrobacterium vector, right? Which is specific genes DNA. It is introduced into the host plant, right? Now the another applications of biotechnology in the field of medicine. So there are several applications in the field of medicine, such as recombinant DNA technology, RDT. It helps for the mass production of safe and more effective therapeutic drugs. The products from non-human sources induce unwanted immunological responses, but recombinant therapeutic does not have such problems, right? When we are using directly the, जैसे कि हम क्या करते हैं, कोई ऑर्गेनिज्म है, उसकी बॉडी में कोई प्रोटीन प्रोड्यूस होता है, बट दैट इज यूजफुल फॉर or that has antibacterial or antiviral property, but we do not directly eat that microorganisms, right? That is harmful. And but we what we are doing in that field of this recombinant DNA technology, we are isolating that particular protein, right, by the DNA sequence, DNA recombinant DNA technology, and we are incorporating that segment of a DNA into the another host and producing that specific protein, and we are taking that specific protein in the form of therapeutic agent, right? Another application, there are different fields of this biotechnology. Genetically engineered insulin is the one of the example of. Gene therapy is another technique which is used in the field of biotechnology. Molecular diagnosis is another an example of application of biotechnology in the field of medicine. First is insulin. So as we know, that is diabetes mellitus. It is the major or largest disorder in the human in India. It is caused due to the deficiency of insulin protein, right? Or our beta cells losing the ability of producing the insulin protein. So what we can do to prevent from that, actually producing that insulin protein out of the natural or the living patient's body, right? In which way we are producing insulin is used to manage adult onset diabetes, right? What is the function of insulin is it maintains the blood sugar level in our body. If it is not produced in the proper amount or sufficient amount, so our blood sugar level get increased and we will be facing a various problem, right? So insulin consists of two short polypeptide chain. One is A chain, another is B chain. That are linked by the disulfide bond. That is, two cysteine residues get the covalent bond between the two cysteine residues of the two chains of the insulin. Next, in mammals, insulin is synthesized as a prohormone, right? It needs to be processed to become mature and functional hormone, right? In the mammal's body, living the humans, it is produced in the form of prohormone. We have to convert it into the functional hormone. So how we can convert, or how we will convert that prohormone into the functional hormone? That prohormone contains an extra stage called C peptide. As you are seeing here, these are the two chains of the insulin protein, which is get attached by the disulfide linkage between the two cysteine residues, right? It is formed or in inactive. It is firstly formed in the inactive form, that is called as proinsulin, right? Now it proinsulin also have a C peptide. This gray color chain is C peptide chain. We have to remove this C peptide chain to convert the pro hormone into the activated or functional hormone, right? So it is removed during the maturation process. Another application of biotechnology in the field of medicine is genetically engineered insulin, right? So in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin. They were introduced in plasmid of E. coli. As you know, the bacteria Escherichia coli having a circular double stranded DNA, that is called as plasmid, which is used as a vector for transferring the DNA of host cell, right? So here we are chain A and B were combined by creating disulfide bond to form human insulin, right? So next is gene therapy. So what we actually doing the manipulation in the DNA sequence of the organism. As we know, there are different types of genes of an organism, such as cystic fibrosis, phenylketonuria, right? These are the disease which is hemophilia, right? These are the disease which is caused due to the single base pair change or deletion or addition of a single nucleotide from the DNA. So which can be curated by this gene therapy process, right? So what is this gene therapy process, right? So what is this gene therapy? It is a method to correct gene defect in a child or embryo. There are two phases of correcting this DNA. One is at germ level, that is at embryo level, and another is at the somatic cell level. So such as bone marrow cells, liver cells, fibroblast. So we are incorporating the change or corrected gene into the somatic cells of the body, right? Here we are not incorporating in the germ cells, because due to the any changes in the human body, so we are not directly incorporating that change into the embryo, right? It is not suggested to do at embryo level, right? We are doing this gene therapy at the somatic cell level, because the somatic cell level gene defect is not transferred to the next generation. If we are incorporating the defective gene into that particular embryo, it is transferred to the next generation also, right? So here genes are inserted into a person's cells and tissue to treat a hereditary disease, right? So the first is germ level gene therapy, which is used as a hereditary, to treat the hereditary disease, which are transferring from the generation to generation. To so if we correct that particular disease at the germ level, that is embryo level, so new forming or upcoming generation doesn't have that disease or disorder, right? So next is one of the gene therapy done in on this lady, that is first clinical gene therapy in 1990 was given to 4-year-old girl with, it has the ability of adenosine deaminase, right, deficiency, which is caused due to the deletion of the gene for adenosine enzyme adenosine deaminase. One base pair is deleted from that enzyme's RNA sequence, right? So this enzyme is responsible for immune system function, right? So if it is not working properly, the immune system is get non-functional, right? So treatment by bone marrow transplantation. What they have did in this gene therapy, they are transferred or transplanted the bone marrow, or enzyme replacement therapy is another type of therapy to cure that such adenosine deaminase disease. That is, injection of adenosine deaminase is not completely curative, but this is not completely curated, right? We can done this on the two ways, right? One is bone marrow transplantation, another is enzyme replacement therapy. If we are replacing the enzyme, it is not completely curing that disease, right? Because we are not actually doing changes at the DNA level or mRNA level. We are changing at the protein level. So in bone marrow, if we are injected that cells with a particular corrected genes, so upcoming forming cells having that corrected base pair, which are producing the corrected mRNA and that are responsible for the formation of correct protein, that is adenosine deaminase, right? Understood? So next is application in medicine, that is gene therapy. How this gene therapy is done? So we have to collect lymphocytes from the patient's blood and grow in a culture, right? The medium we are used to, we have to use the medium here, introduce a functional ADA, that is adenosine deaminase enzyme, cDNA. We are not using the adenosine deaminase enzyme, we are using its complementary DNA into our lymphocytes using a retroviral vector. So retroviral vectors are those vectors which are performing the reverse transcription, right? Like, as in commonly we are forming mRNA from the DNA, but the retroviral vectors have reverse ability. They are forming from cDNA to the double stranded DNA. cDNA is complementary, it is similar to the RNA, right? It is single stranded DNA. cDNA is the single stranded DNA, which is similar to the mRNA. So, what we are doing here, we are using the cDNA and we are converting it into the double stranded DNA by the process of reverse transcription, right? They are returned to the patient. Then after that, we are incorporating that a retroviral vector into that patient's body. This should be periodically repeated, as the lymphocytes are not immortal. So we have to do till the lymphocytes get killed, right? Third is molecular diagnosis. How we can use the biotechnological application for the molecular diagnosis. Here are the conventional methods. One is serum urine analysis are not suitable for easily diagnosis of disease. Various diseases are not diagnosed by the urine or different serum tests. So what we are doing here is we are analyzing, or we are doing the molecular diagnosis. It is possible by the techniques like recombinant DNA technology, PCR, and ELISA technique. What is this PCR? PCR stands for polymerase chain reaction. What actually the principle of this PCR is, presence of pathogen is normally suspected only based on symptoms, right? By this time the concentration of pathogen is already very high in the body, right? However, a very low concentration of a bacteria or virus can be detected by the amplification of their nucleic acid by PCR. We can detect the present different types of RNA sequences by the PCR method, right? Next is uses of PCR for the detection of HIV patients, then to detect the mutations in the genes in cancer patient, to identify many other genetic disorders. A single stranded DNA or RNA tagged with radioactive molecule that is called as probe is hybridized to its complementary DNA in a clone of cells. What we are doing here? We are taking a double stranded or DNA or RNA, we are tagging with radioactive molecule, right? And we are hybridizing it to its complementary DNA, right? In a clone of cell. It is followed by the detection of you, and after that we are detecting the radiography or illuminance light illuminance we are detecting by the autoradiography technique appear on a right. If there is a change or the DNA is get changed or altered, so it will not show the illuminance or light on the photographic film, right? Because the probe will not have complementary with the mutated gene. If we are making complementary, we are binding the we are making a complementation. So if there is no similarity between these two gene sequences, they are not binding to each other. So there is no elimination. So in this way, we are using the PCR, right? Hybridization technique or technique, right? Another technique that is it is by detecting antibodies synthesized against the pathogen, right? So in this way, we can detect the surface protein or surface pathogen by the ELISA test, right? Another applications of biotechnologies is the production of transgenic animal. So here are some examples of the transgenic animals. So what we are did in this transgenic animal, we have manipulated the genome we are incorporated the extra foreign gene in the DNA or genome of that organism and converting them into the transgenic animal, right? Example is transgenic rat, rabbits, pigs, sheep, cows, and fishes. These are the model organisms, right? Which is because the mouse is showing the larger amount of DNA similarity, right? Human genome has 33,000 or 3.3 million base pair, and the mouse is also the mammal, and it is also showing the similarity with the genome of the human. So we are using mice as a model organism to study the various biological process inside that organism. So what are the benefits of the transgenic animals? First is the to study the regulation of gene on normal physiological and development process. So what the physiological process, how this by using this transgenic animal, right? Then another is the study of contribution of genes in the development of disease. So which by this transgenic animal we are able to study the which genes are responsible for causing the disease, right? And which protein is responsible for a specific disease, right? Then what are the biological products produced by the specific genes, right? Then vaccine safety testing can be done by these transgenic animals. Fifth is chemical safety testing, that is to test the various chemical in a body of this transgenic animal. So what they are responding to that particular chemical, right? Another is how we are studying the regulation of these genes on a normal physiology and development. So example is study of complex factors such as insulin like growth factors. These genes from other species that are alter formation of this factor are introduced and the biological effects are studied. Right? We are incorporating the segment of a DNA into the another organism's body, right? Which are having which are belong to the different species, and we are checking the regulation and their physiological role inside the body. We are studying the physiology of that particular gene inside the other species body. What that protein are working in the different species or different organisms body, right? So this gives information about the biological role of the factor in the body, right? Next is the we can also study the contribution of genes in the development of disease by this transgenic animal, which are helpful for studying the contribution of gene. Right? Transgenic model help to investigate a new treatment for human disease. Example: transgenic model for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Benefits of transgenic animal. So in that, biological products can be synthesized. Transgenic animals are used to produce biological products by introducing a genes which code for a particular product. Example: human protein, that is alpha 1 antitrypsin, which is used to treat emphysema, right? It is one of the lungs disease which is related to the respiratory tract, or one of the product which is synthesized, product for the treatment of phenylketonuria, that is PKU, and cystic fibrosis can be obtained from this genetically modified or transgenic animals. Then human alpha lactoalbumin, it is the protein it is nutritionally more balanced product for the human babies than natural cow milk, right? So we are using a balanced diet for our babies, right? Normal cattle doesn't producing that alpha lactoalbumin. We are producing that alpha lactoalbumin in the cattle's body, right? Which is similar to the human milk, right? It have a nutritional value similar to the human milk, right? In 1997, Rosie, that is the first transgenic cow which is produced, and it having a human protein enriched milk, right? 2.5 gram per liter, right? It contains 2.4 gram per liter human alpha lactoalbumin protein, right? Next, next is vaccine safety testing. So as we know, in the early life we have taken various vaccines, right, for resistance of the various diseases like polio vaccine, hepatitis vaccine, right, herpes vaccine. So this vaccine is first tested on the that transgenic animal. So transgenic mice are used to test the safety of the polio vaccine, right? It is, if it is reliable, they can replace the use of monkeys to test the safety of the vaccine, right? First they are doing the test on the transgenic mice, then they are testing the on the monkeys. Then another is chemical safety testing, that is toxicity testing. So these are we have seen. Then another ethical issues are various ethical issues like in the field of that biotechnology, that transgenic animal. So we are completely changing their genome sequence, right? DNA sequence. So their behavior also get changed. which is being unpredictable, and their behavior are unpredictable, right? So it, to maintain such ethical Indian government has set up organization like GEAC, that is Genetic Engineering Approval Committee, which make decision about the validity of genetically modified research and the safety of that genetic modified organism for public service. So this committee is giving approval whether this organism, that is transgenic animals, is safe for public or not, right? Biopiracy, right? So it is the use of bioresources by multinational companies and other organizations without proper authorization from the country and people concerned. Other companies product by without proper authorization. We are not taking authorization from that company or that country, and we are utilizing that product, right? So that is, we can call that is a biopiracy. Certain companies got patents for products and technology that make use of the genetic materials, plants, etc., that have been identified, developed, and used by farmers. Means some of the companies have patent the product such as basmati rice, herbal medicine, that is turmeric and neem. Turmeric has the which is in the thousands of years in the ago, that is given in the Ayurveda, that is, the turmeric has a heavy ability of healing, right? And it is a anti-inflammatory also, right? So this basmati variety rice is also produced in India, but the several companies are unauthorizedly patenting that product. So that is called as biopiracy. Next, it has to develop a laws to prevent unauthorized exploitation of that bioresources and traditional knowledge. Indian Parliament has clearly stated. So what that Parliament has done, they have carried out one bill that is called as Indian Patents Bill that takes such issues into consideration, and including patent terms, responsibility of that organization to be take care of that. So we have seen various applications of biotechnology. Now, as we have before seen, that is gene therapy and cell therapy. So it is the modification or alteration, right? So how we are doing that alteration? So we are genetic material is delivered into the patient atrophy in infants, right? And hemophilia B in adults. So these are the gene therapies done on the different ways. One limitation of current gene therapies is their delivery system, right? So they are typically delivered delivered to the patient via viral vector. So there are the like muscular atrophy in, right? But this approach has typically only affect the cells in the liver. Right? Next is the synthetic biology. It is one of the trends of the biotechnological field, that is synthetic biology is, it is the involves the whole. We are
Nice, sir. We are not getting his voice.
अचानकच आवाज गेला सर.
globe.
सर गुरु सब्जेक्ट खूप चांगला होता पण आवाज नाही येत त्यांचा.
नमस्ते सर. सर आपने गोल्डन राइस की बात की है और विटामिन ए का जिक्र किया आपने. क्या विटामिन B12 गोल्डन में मिलता है हमें? खान सर आप अनम्यूट होइए. म्यूट है आप. मेरी आवाज आ रही है सर?
आपकी आ रही है. खान सर को नहीं जा रही आवाज. खान सर वो माइक अनम्यूट कीजिए.
यस. हाँ बोलिए सर. बोलिए सर.
यस सर. यस. जयवीर सिंह सर, नाउ यू आस्क द क्वेश्चन.
सर मेरा सवाल है, जो आपने गोल्डन राइस की बात की है विटामिन ए की, क्या विटामिन 12 B12 इसमें राइस में गोल्डन में होता है? और हाँ जी
बोलिए. और कुछ? यस.
मेरा सवाल ये है सर, अगर हम हाँ दूसरा सवाल भी बोल देता हूँ. जो हम बायोटेक्नोलॉजी से क्रॉप में वृद्धि करते हैं और जो फूड्स को हम यूज करते हैं, उस फूड्स में और हमारे नेचुरल फूड्स में कौन सा अधिक उपयोगी है?
सर, जो पहला सवाल है आपका विटामिन B12, सो जनरली जो गोल्डन राइस है, जो उसमें हमने मॉडिफिकेशन ये किया है कि कौन से चीज को हम स्पेसिफिक चीज को जो है तो उसमें इंक्रीज करा रहे हैं, राइट? तो स्पेसिफिक चीज हमने टारगेट किसको किया है? विटामिन ए को टारगेट किया है। सो विटामिन ए को टारगेट किया है तो उसका अमाउंट को हम इंक्रीज करा रहे हैं, बट इट स्टिल हैव small amount of vitamin B, but उसकी कौन ने क्या किया है? To avoid the deficiency in the deficiency of vitamin B in the which is happening in the developing countries, right? Like we know the vitamin A is contained in the carrot, right? And different milk products, right? So they are not affording that milk product. The poor people can't afford that milk products and carrots, right? So to avoid their problem, that scientists have developed the vitamin golden rice, right? यस सर, उन्होंने क्या किया है, जो हमारी डेवलपिंग कंट्रीज है वहां पर जो पुअर पीपल रहते हैं, तो उनकी जो है तो उनकी इतनी कैपेबिलिटी नहीं रहती है कि वो बहुत सारे जो है कॉस्टली चीजें जो है तो खा सके, वो जो है ना फूड मटेरियल जो है तो परचेस कर सके। तो उसको अवॉइड करने के लिए उन्होंने क्या किया है कि ऐसी वैरायटी बनाई है राइस की, दैट इज known as golden rice, and have also made a golden mustard also. That golden color is due to the vitamin A, right? विटामिन ए की वजह से ही उसके अंदर जो है तो गोल्डन कलर आया है, राइट? सो होता क्या है, जो आई नाइट ब्लाइंडनेस का जो प्रॉब्लम है, इट इज ड्यू टू द डिफिशियंसी ऑफ विटामिन ए दैट इज बीइंग काउंटर्ड बाय द दैट गोल्डन राइस, राइट? और सर आपका जो सेकंड क्वेश्चन था, जो हमारी नेचुरल वैरायटी है और जो हमने जेनेटिकली मॉडिफाइड किया है, तो कौन जस्ट उसको जेनेटिकली मॉडिफाइड कर रहा है। अब हम जो चीजें खा रहे हैं जनरली यूजुअली हमारी जो डेली टू डे लाइफ में, वो नियर अबाउट मोर देन 30% वी आर यूजिंग द जेनेटिकली मॉडिफाइड क्रॉप्स, राइट, इन आवर डेली लाइफ, बट इट इज नॉट हार्मफुल, right? Our natural are the very good, but due to produce in small amount large nutritional quality food product. So that's why we have to do such things, right? We have to modify that plant and animals to increase their productivity in the nutritional value and to increase the nutritional value of that, right?
धन्यवाद सर, बहुत बहुत धन्यवाद।
Is there any question?
Good evening, sir. Good evening, Dr. Getkar sir, Principal sir, Prashant sir. Good evening. And good evening, sir. Thank you very much. Wonderful presentation you have given for this biotechnic and biological. I will consider it as a bioengineering also. Wonderful presentation. You have talked about various plants, and then we talked about various, we can develop genetic structure, composition, we can change in animals also. And we can use this technique for therapeutic purpose, preventive purpose also. So wonderful presentation, sir. Thank you very much. My basic question is, it is regarding, vegetables. Like nowadays we are getting carrots and radish and other things. They are in a good in size, but poor in taste. And most of them are reconstructed or something like that. And hybrid, what we use a word for that. And it has no much taste as such. Muli pehle tikhi lagti thi. Nowadays you'll find muli, it appears, reddish appears like a coconut. And even, the vegetables like brinjal has no taste at all. Right? That has more, sense. So whether we are increasing just productivity and we are losing the basic essence, it may be one of the side effects what I feel. So what you suggest about this, one thing. Second thing, yes, you correctly said, nowadays in a disease conditions, definitely we can use this technique because there are so many diseases, immune system related diseases, the one's body fights against their own tissue. So in these instances, yes, definitely this technology is wonderful. What you had talked about, diabetes mellitus, yes, definitely over there also we can play the vital role. And no doubt, in various places like cancer and other where we are not having any answer, we have to just wait and watch for the patient to get terminated. Even we are doing chemo and radiation therapy, everything, but it doesn't, leads to a positive life. So this is one of the answer, we can search for in this biotechnology. That is a wonderful technique, what you have said. Correct. That day will not be long, sir. You will find the husband wife goes to doctor and they will ask, we want this kind of kid. The height should be like this, the height should be like this and that. And then, the product will be coming like that only. So that genetic structures and genetic engineering is playing a wonderful role, and that will come definitely. I'm very sure within 2 decades, it will come that we will have a child, what they want. Right, and we don't have to ask, kis pe gaya hai, right? Daddy pe gaya ki mummy pe gaya. We don't have to ask for that also. So these are the new advanced technologies. Definitely, it will play a positive role also. But to some extent, I feel that the natural essence, will get lost when we are going for such a kind of advancement. So, what do you think about this, sir? Thank you.
Sir, as you have told,
सर आप वीडियो बंद करके आंसर दीजिए जिससे आवाज क्लियर आएगी. हाँ जी.
Test in the natural foods are ठीक है सर. मेरा तो दिखा रहा है पर सर सर आपका जो क्वेश्चन था कि जो नेचुरल फूड है हमारी उसका टेस्ट अच्छा होता है और जो हमारे पास फॉर एग्जांपल हमको घर में कनेक्शन लॉस्ट हो गया. हेलो
Sir, there may be network issue. Principal sir. So thank you so much, Honorable Jaivir Singh sir, Dr. Swanand Shukla sir, for the nice queries. Due to technical issue, our voice can't reach to Honorable Khan sir. So thank you so much. Again, Dr. Swanand Shukla sir, Jaivir Singh sir, if you have any query and couldn't be analyzed it clearly, the voice couldn't reach towards you, then you ask next time with the contact given.
Sir, there is a network problem. Please. Hello.
Sir, now I'm moving towards the vote of thanks.
Yes.
If you would like to, if you got the question, please answer, sir.
Yes.
You got the question from Dr. Swanand Shukla sir?
Sir, I have not listened because there is a network problem. So it is, I have rejoined. Yes, yes, yes. अच्छा, अच्छा. हम्म, हम्म, हम्म.
Thank you, sir. Now I move towards the vote of thanks. Shri Kumarswami Mahavidyalaya
Thank you.
Ausa, District Latur. As we know, organized World Record Series 2. Today we have covered 240th session of the series. And the keynote speaker for today's session was with us Honorable Khan Zahid Iqbal sir from Aurangabad, Maulana Azad Arts Science Commerce College. And the sir have talked about the topic biotechnology trends and its applications. Now this topic have presented simply in a simple language where we understood a lot from your lecture, sir. Really, it was very wonderful session. The information given is was very informative for us. So thank you so much on the behalf of organizing committee members. I thank the chief organizer of the series, Honorable M. Bechkar, sir. Also thanks the faculty members from my college, Dr. Anita Dole, madam, Honorable Praveen Udge, sir, Honorable Dr. K. N. Kadam, sir, and remaining all others. Then I special thanks JV Singh sir, Dr. Swanan Shukla sir, who always join the series on time and ask nice queries, also share some views from their sides, and heartily thanks all those dear participants. Now I request you all please fill the feedback link which is given in the chat box. So with the kind permission of the chief organizer, I declare today's session is over. Once again, thank you very much, Khan sir, for your wonderful
Thank you, sir.
presentation and being with us, spare your valuable time to us. Thank you very much.
Thank you. Thank you all. Thank you, sir.